Hello and welcome to this course. I am Dr. Sriram Kumar. Today we are going to learn the second part of biological methods of nanomaterial synthesis. In last class, I discussed the nanomaterial synthesis using two key biological systems, that is bacteria and the fungus. So in last class, we understand the reason behind the synthesis of nanoparticle by the biological system, specifically by bacteria and the fungus, and that is the detoxification. And we understand the reason and the process behind the detoxification. So there are the four different kinds of the process that occurs in the detoxification process, and that are the reason of the synthesis of nanomaterials by biological systems. Then after we understand this mechanism of nanomaterial synthesis by intracellular mechanism and the extracellular mechanism. So here, extracellular mechanism happens on the cell wall or maybe the cell membrane that depends on the kind of organism, biological system we use. And there is the other kind of the mechanism that is the intracellular mechanism, and that occurs inside the cytoplasm. And we understand with the example of the synthesis of zinc oxide nanoparticle using NADH kind of the enzyme. And we also understand the kind of extracellular kind of synthesis of the zinc oxide nanoparticle using extracellular mechanism. And then after we understand the difference between these kind of the mechanism, extracellular and intracellular kind of the synthesis process. And then after we understand the most important things of the synthesis of nanomaterials by the biological system. The advantage of using the biological system is that we can synthesize nanomaterial with the monodispersity. That means we will have the control over nanomaterial size. At the same time, we can control the size of nanomaterials. So if you compare the nanomaterial synthesis before and after, we have the huge difference in the size and polydispersity of the nanomaterials. So we have some kind of the controlling parameters that are the pH, temperature, cell is concentration, reactions times, radiation, and stirring. So I discussed all these parameters in detail in the last class. So if you are looking the effect of these parameters, go there and watch again that video. And these are the references if you are looking the factors that can affect nanomaterial synthesis by biological system. So here I put here the three references. One is for the gold nanoparticle and those two are for the silver nanoparticle. And then after I put some references so that you can take that references for your application, or maybe the synthesis of nanoparticle by this bacteria, maybe this fungi, algae, or maybe the plant kind of the system. And then after we understand the kind of the nanomaterial synthesis using fungi, and we understand why we should use fungi as compared to bacteria for nanomaterial synthesis. And we understand with the three different points. That is, one is that is the effective secretion of the enzyme, that is the extracellular kind of the mechanism. And second is kind of the high production of the enzyme. And the third is kind of this metal bioaccumulation's capability of in fungi is better than the bacteria. And we also understand with the example of how the fungi also takes part in the synthesis of material using the intracellular and extracellular kind of the technique. So this is the example of the gold nanoparticle synthesis. And similarly, we have the kind of the intracellular and extracellular mechanism that depends on the mechanism process. And we also understand kind of the complex redox species present here. So this kind of the process happening here, so inside the plasma, that is because of the redox species present, and because of that the redox species and the enzyme, so there is some kind of complex reactions, and based on that we'll get the nanomaterial synthesis from M plus to the M. And we also understand with example we with the fusariums, oxysporum kind of fungus. And then after we also understand the different kind of nanomaterial synthesis using the extract. So till now we understand, okay, if we take culture fungus or maybe this bacteria, so that nanomaterial synthesis can happen intracellularly or maybe the extracellularly. But we can also extract some useful chemical, enzyme, protein kind of the things from that biological system, and we can use that chemical in synthesis of nanomaterials. For example, in this case, from the fungus, we can extract some useful chemical, and if you just put this kind of the nanometal ions in that from the extract, we will get the kind of the nanomaterial synthesis, and that nanomaterial is capped and that is stabilized by this kind of the fungal extract. and uh, and then after we understand difference between the fungus and the bacteria mediated gold nanomaterial synthesis, and we understand that the biosynthesis rate of the gold nanoparticle is much higher as compared to the bacteria, and therefore there is a kind of scalability of fungus at the industrial level as compared to bacteria because of kind of control over the size of the nanomaterial, at the same time the kinetics rate of the synthesis of nanomaterials and as compared to the bacteria. Okay, so today now we are going to learn nanomaterial synthesis using protein. So we are going to use protein as a template to synthesize nanomaterial. So now let's take example of the protein, and here that we are going to use the ferritin protein. Ferritin protein is very important for all animals because that ferritin protein is works as the kind of the that is stores iron in our body. What happens? The ferritin protein known as the storage of iron in our body. So whenever there is this deficiency of iron in our body, so that protein provides a supply that iron to body, and that case we and that prevent kind of the disease cause of this deficiency of the iron in our body. So now let's understand that is important protein. So if you want to understand the structure of this ferritin kinase protein, so if you understand the outer size of that protein is 12 nanometer size, and that and inside this protein, if you just remove, so in that cavity there is the iron inside the cavity. So if you just take out that iron from that cavity, so what happens? So we will get kind of this cavity, and that is from 6 to 8 nanometer. So it depends on. So for the human ferritin, so the size of the that cavity is close to the 8 nanometer, and in some other organism, the size may be the 5 to 6 nanometer. So here, this portion is kind of this protein shell. This is cavity, we can say, and these are kind of the channel through which the metal ion can enter inside the cavity. So now let's understand with a visualization that how protein looks like. So this is kind of the external structure, and that diameter of that diameter from here to there is 12 nanometer, and the internal diameter is 8 nanometer. So we can exploit that internal diameter of that protein, and we can put here some kind of the nanomaterial there so that we can use that kind of the nano enzyme. So that is kind of the artificial enzyme. So that can be can or at the same time, this specified size of the internal structure of this protein can be used to synthesize the size control synthesis of nanomaterial. At the same time, we can also modify the surface of the nanomaterial with the specific kind of the chemicals. Now let's understand the synthesis of nanomaterials using ferritin protein. So what happens here is, so here this kind of protein is called the ferritin protein, and when you will be take out iron from inside the protein, because iron is normally present inside of the protein. So this structure is called FO, ferritin, kind, or we can write it AFT in short form, ferritin kind of structure. So we can use that structures to synthesize the nanomaterial. So here, so this is the shell here, ferritin shell. And if we use kind of this metal ion, suppose if we use the metal ion, for example, gold plus 3, maybe kind kind of things here, maybe the gold and the silver plus here. So these metal ions enter through the channel inside the ferritin protein, and in the presence of reducing agents. So here, so that metal ion enters inside the ferritin, and if we add here so reducing agent, so what happens? So that metal ions get reduced. So what happens? We have Amen, and we get the some kind of the M not M0, and from this one there kind of the nucleation process happen, nucleation process, and from there is kind of the nanomaterial formation, nanomaterials formation. So these are the steps of the synthesis of nanomaterials. So in the presence of the reducing agent, so these metal ions get reduced, and we have the such kind of the nucleation for the growth of the nanomaterials. We have here the nanomaterial nanoparticles inside ferritin, and this is kind of the encapsulated with this protein, and that makes this one is the biocompatible in nature, and we can use this one for the different purpose or different application that depends on the further functional modification. So now let's understand with a different example how to use this protein for the synthesis of maybe cadmium selenide quantum dot. So here we can for again exploit the size of the cavity of ferritin protein. So what happens if we add here cadmium plus 2 in the solution? What happens? So because of the uh, channel, so that will enter inside this cavity, and here we can use EDTA to control the migration of the cadmium plus 2 from outside to the inside, because this cadmium and EDTA will make complex, and that will hinder the diffusion of this cadmium EDTA from outside to the inside. So here the EDTA is acts as the kind of the complexing agent. So here EDTA acts as the complexing agent. So what happens? So it controls diffusion or diffusion of cadmium 2 in cavity. And if we have the control over the diffusion of the cadmium in the cavity, we can control the growth of nanomaterials. So that help in the growth of nanomaterials. So these are the things is happening in the presence of ADTA. And if we add here the precursor of the selenide, so that will enter inside, and there is the formation of the cadmium plus 2 and selenide, and we have the kind of the cadmium selenide. So there is the kind of the nucleation process happens, and then after that is for the growth of the nanomaterial. And once we do the dialysis in the presence of kind of the things, so that we can do the dialysis process in, so here this dialysis process we can do in the thioglycolate. So if you use the thioglycolate kind of the chemical and dialyze, so this is can be used as a kind of the capping agent. Further, if you want to modify, and or maybe since we have already used here cadmium ADTA to control the diffusion, so it is better to not use for the thioglycolate here. So here we can use only dialysis purpose. We can only use here kind of the dialysis, maybe the sodium acetate. So we can use here sodium acetate. So in presence of sodium acetate, so unreacted precursors, maybe the selenide or cadmium ADT, that complex comes out, and we will have kind of the encapsulated cadmium selenide, this kind of this nanomaterial. So we can use these nanomaterials for diverse applications. So now let's understand. So till now you understand the example of how to synthesize nanomaterial using that cavity of protein. So here the mechanism we can so there may be the two kind of the mechanism. So what is here is that, so this one, so this is a ferritin where we have here iron plus 2 in the cavity. So first we need to take out that iron plus 2 from that cavity. So once we take out and we have this here's cavity there. So this structure is called apoferritin. So ferritin is kind of protein which has iron plus 2 in its cavity, and if we take out that iron, we get kind of protein structure that is called apoferritin. So once we get this kind of the cavity, then we have the two kind of approaches. One is kind of nanoreactor. So nanoreactor means we are exploiting the nano cavity inside the protein. So in this case, if we just use the precursor there, what happens? So that will diffuse inside through the channel, and in the presence of reducing agent, we will have a kind of the nanomaterial synthesis, right? So here, so in this process, we are exploiting this cavity inside protein, and that is called the nanoreactor. And the second kind of the approach is kind of a reassembly and the de-assembly process. So what happens here? So up to the apoferritin, we can play with the kind of pH, and based on the pH, we can change. So we can use kind of the assembly and de-assembly process. So we can use here the assembly process. So this assembly process can happen if pH is close to 2. And that the process happens, the assembly process happens when the pH is, so here, if that pH, so if that pH is greater than 5, so it was the more pH, so there is kind of the reassembly formation. Here the pH is greater than 5. So what happens here is we can do here is we can add here, and we can add here the reducing agent at low pH. So what happens? We will have kind of the formation of nanomaterial. And in that nanomaterial, there is kind of de-assembly process happens. So this is kind of the de-assembly of the apophilitin kind of protein. And once we just increase the pH, so these kind of the fragmentations can again reassemble, and there is kind of this nicely quantum dot encapsulated inside the protein. So we can either exploit that nanoreactor kind of this technique, or maybe we can use this de-assembly or maybe reassembly route to synthesize nanomaterial. We can also understand with other example that if we can synthesize platinum nanoparticle here. So if we have precursor of the platinum here, so at pH 8.5, what happens? We have kind of the cavity here, and that cavity is the 8 nanometer. So that precursor will enter inside the cavity of the protein, and and this is the NABH is acts as the kind of reducing agent. So in presence of NaBH4, so here platinum plus 2 get reduced to just platinum 0. And that is the mechanism. So what happens? So this is kind of the platinum FTN means, so this is kind of the apoferritin. So this is kind of the platinum nanoparticle modified with the apoferritin. So we can either synthesize nanomaterial using of the platinum nanoparticle, maybe cadmium selenide, if we can synthesize. So this is example of the lead sulfide kind of nanomaterial. Here this is example of cadmium selenide, and this is example of the platinum gold silver nanoparticle. And if you want to further synthesize nanomaterial of the cadmium selenide, so this is the example of lead sulfide. So we can use similar mechanism here. So in this kind of the approach, we should convert ferritin to apoferritin by removing iron plus 2 from this one. So we get the apoferritin, and we titrate the cavity with NaCl. And when we add here the cadmium acetate, that is the source of cadmium plus 2, so that cadmium that will enter inside the cavity. And if we add the sodium sulfide in 2s, what happens? We have cadmium plus 2, and we have sulfide, and that forms cadmium sulfide inside the cavity. So this is the kind of the synthesis of the cadmium sulfide. And then after we do the dialysis, so after this one, we need to do dialysis process, and then we get the kind of ferritin modified cadmium sulfide nanoparticle. So this is all about the kind of this protein encapsulated or the protein templated nanomaterial synthesis. So if we use the protein kind of the encapsulated nanomaterials, so this kind of the synthesis process makes the more biocompatible nanomaterial, right? Because the surface of the nanomaterial is modified with the protein, so that makes them more compatible in nature. So now let's understand the use of plant extract in the nanomaterial synthesis. And that is very important aspect of the synthesis of nanomaterial. So now let's understand. So here plant and a plant extract. So that provides the single steps biosynthesis process, and this protocol involving free from toxicants and a natural capping acid, and can generate biometallic silver and the gold shell nanoparticle, excellent stability and size control, cost-effective, large-scale production of bimetallic, And we can synthesize different kind of the nanomaterials, maybe this metal oxide, maybe this semiconductor kind of the nanomaterials. So now let's understand how we should use plant extract to synthesize nanomaterial. So what happens here is if we somehow extract this very important key aspect of active material from the food waste. So what happens if we have the food waste? So from that food waste, we can extract the important chemicals from that plant and they earn from that food. And so in kind of this food, we will have some, maybe we have, so here, what kind of things we will have here? So in this case, we'll maybe have the kind of the important enzymes. Maybe we have some kind of this proteins. Maybe we have kind of the polysaccharides. Polysaccharides. Maybe we have the kind of the alkaloids, kind of the chemicals. So we can extract these important chemicals from food waste. And when we add here this metal, metal salt solutions in that extract, so what happens? So here, because these acts as a kind of the natural reducing agent, so these acts as a kind of the natural reducing agent. So in presence of natural reducing agent, so here this metal plus get reduced and we'll have this one and this is this to the nanomaterial. Nanomaterials. So this is the mechanism. So what happens here? We have metal salt here, and if we just extract from the food, what we have this kind of the active chemicals, and what happens? So that reduces metal plus to here the M0, and so this is kind of the nucleation steps. And if that for the grow, we have the kind of this nanoparticle. And in that food, we have some kind of the stabilizing agent. So that stabilizing agent maybe here, maybe example of the enzyme, functional group, maybe the protein acts as the stabilizing agent. And then that functionalizes the surface and that stabilizes nanomaterials. So this is the kind of this mechanism using the plant extract. And either way we can understand these things. We have the plant extract here, and if we use just add the metal and source there, so we'll have this process. So we have the kind of manipulation process, then for the kind of the growth, and there is the stabilization and the capping agent by the kind of protein, maybe enzyme kind of things. So here we can control the synthesis using the different parameters, maybe the pH, maybe concentration, reaction time, and the reactions temperature. I already explained these kind of the parameters. So we can control here nanomaterial synthesis. So till now we understand the synthesis of nanomaterials using biological system. We use bacteria, we use fungus, then we use protein as a template, and we use plant extract to synthesize nanomaterials. Now let's understand the difference between the biological synthesis and non-biological synthesis. So here, biological synthesis here, optimization of design of the parameters. So in biological synthesis, we need to optimize different things, such as we need to optimize the kind of the organism we need to use. Second is kind of the cell growth condition. So we need to use always the growth condition of the biological sample growth phase. However, in non-biological system, we need to control over the physiochemical conditions such as the reaction conditions, catalyst, concentration of the reactant. However, in the biological system, we need to consider the choice of the organism, maybe the bacteria, fungus, or maybe we should consider the protein and this growth condition of the cell. And what is the mechanism of the synthesis here is that, so in the biological system, maybe there's enzymatic and non-enzymatic process. So whole cell or cell extract. So in the biological synthesis, either we can use whole cell or maybe the extract from that biological system, and then we can reduce metal ion into this metal. However, in the non-biological system, there is kind of the chemical reductions, irradiation, pyrolysis, electrolysis, or Sol gel process. So these are the techniques in the non-biological system. And on the parameters with the uses, so we can use the biological system in the kind of the therapeutics, clinicals, biomedical, and the diagnostics fields, such as targeted drug delivery, antimicrobial agent, magnetic resonance imaging. So this is kind of the MRI and biosensors kind of things. And here in non-biological synthesis, so this is restricted in the clinical and the biomedical fields due to stress of the toxic chemicals. And if you think about the kind of this environmental concern, so here biological synthesis is the kind of the eco-friendly and that doesn't contribute in this pollution. However, the non-biological systems may maybe the may cause the pollution, maybe soil, water, due to the toxic chemicals. And the duration of the synthesis is that, so here the non-biological system requires the less time. However, the biological system requires a longer time because the growth of the some microbial growth. So for the enzyme, also for the kind of the fungi, we need close to the 1 hour, and for this bacteria, maybe we need more than 24 hours. So it depends on the kind of the system we are using. However, in the chemical techniques, we require very less time. So this is all about kind of this nanomaterial synthesis using biological system. And in next class, we will learn biotemplated nanomaterial synthesis. So we will use different template, and then we can modify that template to synthesize different shape and size of the nanomaterials. And then after, we will learn bio-inspired nanomaterial synthesis. So these are very, very important topic in biomaterial synthesis. And so see you in the next class. So we will have we are going to have very important technique and understanding about the nanomaterial synthesis using these two technique. So see you. Thank you.